Hello everyone, this is Miss D.M. Alvekar working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. And in this lecture, we are going to study very interesting topic, that is glycogenolysis. In every day, we consider or we remember these terms by breaking that term into the two different terms. So here also this glycogenolysis, lysis means breakdown, and glycogen is the carbohydrate, type of a carbohydrate which is a polysaccharide. Now why there is a need of the body to store that glycogen? Generally in our body the glycogen is stored in the form of small granules at liver and the muscle cell. Now why there is a need of the body so that it can synthesize the glycogen in the form of a granule, or it will store that glycogen in the reserve form or the storage as a storage depot. Because whenever we eat, the number of a glucose molecule will get increase in the blood, and then when there is a no any demand for that glucose, then that glucose if remain as it is, then it is the hygroscopic. So there is a body will not keep that glucose as it is for the longer time. So that glucose has to be stored in the different form so that it will not absorb so much water, and in future it can use whenever the body will demand. So the glycogen is the best form of the storage form of a polysaccharide in the animal cell. Especially it is stored in the liver and the muscle. So when this glycogen is used, now it is used whenever there is a fasting condition or whenever you are performing the exercise, then at that time during exercise condition, when the body will demand the glucose, then that supply is made by utilizing the glycogen which is present in the muscles. But whenever the body is at the fasting condition, at that time the body will utilize the glycogen which is stored into the liver. So let us begin about the how the glycogen is broken down into the glucose molecule. Because we have already studied that whenever the demand of the energy is there, which is in the form of a ATP, and that ATP is synthesized or produced by the process of glycolysis, Krebs cycle, and oxidative phosphorylated product, that is NADH, FADH2, that are also giving the ATPs, but that has to be transported to the electron transport chain. So glycogenolysis will play a very important role in order to maintain the blood glucose level. So whenever there is a fasting condition, the glycogen is broken down into the small glucose units molecule, and the demand will get fulfilled. But whenever there is a demand of the energy by the muscles, at that time the glycogen is broken down into the glucose at the muscle level, and then that glucose will be transported or it will enter in the glycolysis and then further the in the Krebs cycle, so that it will fulfill the demand of the energy during the exercise. Now as we know, if the blood glucose level will get increased, then the insulin will get secreted by the pancreatic beta cell and the blood glucose level is get controlled. But when the blood glucose level will get decreased, then the pancreatic delta cell will secrete the glucagon hormone, and then glucagon hormone is the first which will stimulate or it will indicate that the blood glucose level has decreasing. So then body has to take care that the blood glucose level will be controlled by using a suitable process. So what is that suitable process? It is nothing but the glycogenolysis. At that time during fasting condition, the body will prefer the pathway of the glycogenolysis. Now when you will observe the structure of a glycogen, the glycogen is made up of a number of monomers or number of monosaccharides which are joined with each other by the glycosidic linkage. Now glycogen is the polysaccharide. It is a homopolysaccharide and it consists of the two different types of a linkage, that is alpha glycosidic linkage and beta glycosidic linkage. Here I have shown you the two different types of bonds, but that is not related to the glycogen, but it is related to the disaccharide. One is a maltose which consists of the two glucose molecules which are joined together by alpha glycosidic linkage. But when you are observing the structure of a lactose, it is having the beta glycosidic linkage. So in the polysaccharide, that is glycogen, the two types of bonds are present in the alpha form and in the beta form. Now what happens, or what are the main reasons so that body will prefer the glycogenolysis? So it is a cascade mechanism through which it is communicated to the liver or the muscle cell so that it will carry out the glycogenolysis process. Now this epinephrine hormone will be released by the adrenal gland in the fear and fight mode of the body, and then that epinephrine is transported towards the storage depot, that is in where the glycogen has stored. Epinephrine hormone will not transfer to this cell membrane, it will just remain on the cell membrane, and whenever it will remain on the cell membrane, it will communicate its signal inside this cell. So how it will transduct its signal? So this is the signal transduction mechanism, where the one molecule will activate another, it will activate further another molecule, and the mechanism will get continue for the activation of the process. So here epinephrine hormone will bind to the specific receptor on the cell where the glycogen has stored, and that special receptor is called as a beta adrenergic receptor. Now this beta adrenergic receptor at inside is attached with the three different subunits, that is alpha, beta and the gamma. This alpha, beta, gamma three subunits are nothing but the units of the G protein. Now when the G protein is in the inactive form, it is attached with the GDP. But as soon as the epinephrine will bind to this receptor, this G protein will get activated, and this alpha unit, it will get separated out from these three units, and then it is activated with the help of a GTP. Now GTP along with the alpha subunit is a charged one, and it will attach to the adenylate cyclase enzyme. This adenylate cyclase enzyme will convert ATP into the cyclic AMP. Now cyclic AMP is the secondary messenger, that is it will transfer the message which is communicated by the epinephrine, which is the first messenger or primary messenger, and that signal has transduced via this G protein, via this adenylate cyclase up to this cyclic AMP. Now this cyclic AMP, which is a secondary messenger, it will activate the inactive protein kinase A into the active protein kinase A, and then this activated protein kinase A will activate the glycogen phosphorylase enzyme, and this glycogen phosphorylase enzyme is present in the two different forms. One is of glycogen phosphorylase A and another is the glycogen phosphorylase B. Glycogen phosphorylase A is the active form of the enzyme which will catalyze the process of a glycogen lysis. So when the protein kinase enzyme will activate glycogen phosphorylase, it will break down the glycogen by the non-reducing end so that it will remove glucose molecule in the form of a glucose 1-phosphate. Here this is the simple mechanism, where suppose this is the small chain of a glycogen which is made up of a number of glucose molecules which are joined together by the alpha glycosidic linkage. This linkage is a alpha glycosidic linkage, and there are number of branching points. So this branch is called as a, or this branch is a formed by the linkage of this glucose molecule with this glucose molecule by the alpha 1,6 type of a linkage is present. So number of branches are present in the glycogen polysaccharide. So here in the process of a glycogen phosphorylase, the activated glycogen phosphorylase enzyme will always act on this glycogen from the non-reducing end. So this is the non-reducing end where you will not observe any free form of aldehyde or the ketone. So it will cut this glucose molecule and convert that glucose molecule or release that glucose molecule in the form of a glucose 1-phosphate. Again, it will again act on this branch of alpha 1,4 linkage, and it will cut that glucose molecule into the glucose 1-phosphate. Similarly, it will cut this linear segment of the number of glucose molecule, and it will generate the number of glucose 1-phosphate molecules. But when it will reach to the 4 units of the sugar from the branching point, it will remain as it is. It will not cut further. Then this step will acted upon by another enzyme, that is debranching enzyme, or also called as a glucotransferase enzyme. The name itself indicates there is a certain transfer. So here these units of the glucose near to the branching point will get transferred or joined to the main chain of this glycogen, and then only one sugar will remain as it is at the branching point. So this is the branching point, only one sugar is remaining, and then this alpha 1,6 linkage is broken down by another enzyme, that is glucosidase. So apart from the glycogen phosphorylase enzyme, the glycogenolysis will require 2 types of a branching enzyme. One branching enzyme will just transfer the branch from sub branch to the main branch, and another branching enzyme will remove the point or the junction of the branch. So similarly, now this chain will again converted into the linear form now. So again it is acted upon the glycogen phosphorylase unless and until it will not reach such type of a branch, and in this way the glycogen will get degraded with the help of a glycogenous phosphorylase and 2 debranching enzymes, and glucose 1-phosphate molecules are released. Now when this glucose 1-phosphate molecule is released, then it is converted into the glucose 6-phosphate, that is the phosphate which is present at the first carbon of the glucose. It just transferred to the 6th carbon of the glucose. It is just catalyzed by the phosphoglucomutase enzyme, and this glucose 6-phosphate will be acted upon by the enzyme phosphatase, that is phosphatase enzyme will always remember the phosphate. So this phosphatase enzyme will remove this phosphate and convert the glucose 6-phosphate into the glucose, and now the demand of the glucose or the level of the glucose will get increased when this glucose is transferred to the blood by this glycogenolysis process. And if there is a demand of the energy by the body, then whatever glucose is released into the blood, that will reach to each and every corner of the body, and then there that glucose will enter into the cell. It will proceed for the glycolysis, then to the Krebs cycle, and whatever the NADH, FADH2 will be generated, that will be carried towards the electron transport chain, and substrate level phosphorylation will give the direct ATP, and oxidative phosphorylation type of oxidative phosphorylation will generate the NADH, FADH2, which will again transport towards the electron transport chain. So similarly, the demand of the body during the fasting condition or during the exercise, when there is no external supply of the glucose is there, at that time the body will use this storage form, that is in the form of a glycogen. So I hope that you have understand this mechanism in very simplistic manner. We have studied about the glycogen, we have studied about the role of the glycogen phosphorylase enzyme, how the cascade mechanism will take place so that the glycogen phosphorylase will get activated and the glycogenolysis process will get produce the glucose 1-phosphate molecule, and how the glucose 1-phosphate molecule is converted into the glucose, so that the demand of the ATP or the demand of the glucose of the body during fasting condition or during exercise will get fulfilled. So thank you for your patience listening. Thank you so much.